and TGF-beta signaling pathway that are the metabolic pathway. Okay, as I haven't yet show to you this information, but the clinical lab data have created to us a big problem, because when we attempt to link this data to clinical data for the same patients, we have a lot of different records. There is not a standardization. Then, for instance, and the method used by the clinical lab to make the standard clinical screening are not always the same. Because if one hospital uses a method A, you have a specific data, a specific response. If another hospital uses for the same biochemical test another method, the response could be different. As an example, my sister make a screening for thyroid hormones in a private laboratory, and given her results. When she gone to the endocrinologist have some doubt about the results and send her to the hospital lab. The hospital lab has done the analysis with another kit, and the results are different. Then, in terms to proceed with the data warehouse in this case, the interaction with the clinical infrastructure is important, and also is important to verify the quality of test. Happened to myself that I made to a screening. I go the first time to the same hospital for this analysis, and they ask is a urine analysis. They ask to me a liter, and I am going in another hospital in my town to do the same analysis, and said no, it's with 1 liter of urine, and said what it is this? I need only a 2. We're talking about 2 different tools, 2 different things in 10 kilometers. Then what is the message in this thing? The message is, if the clinical go to you information about the exposure, the first thing that you must to ask is, okay, let me know what is the instrument. How many blind control or cross control has been done. Otherwise, Professor Pazzardani said, but what we have done, the performance is so low. No, it's not that you can say, okay, on my test domain is working very well, but now on this clinical data, on the biochemical data, it doesn't work. Be careful with the clinical data. Clinical biochemical data. At the moment, now, this database contains 10,000 records of patients. Then the idea to rescue, I have a file of Excel that is too big. It's too big. But if I have more cell empty than fill, I'm not so sure to use any machine learning tool or clustering or what you want to classify it. This is a big problem. In a theoretical way, you can use it, genes, SNPs, and go in the qualitative database. Okay, I would like to come back a few minutes on the back on this slide, because in fact this focus what I am doing now and what are the problems. About in this is an attempt to link almost the molecular aspect of monitoring. When I start with a bilateral project with NIRI, we must attempt to make the backward association between protein and microRNA. Then after we are changed to analyze the exposure, the chemical, the PM10 chemical identification. In this case, for instance, the problem, there are additional problems, for instance. Our chemicals can modify some processes inside the cell that not directly appear in the phenotype, but on long term appear in phenotype. Then the problem and the first problem is that many, as I said, that many chemicals, that many studies on PM10 consider PM10 as a unit. Only few studies, and only now start to give information about the quality of pollutants inside this class of mixture. The other thing is that the possibility that compound can target more than one genes, more than one protein. In this case, for instance, in which way one can collect and can put in a data warehouse the relevance or interaction. In this, the basic idea is the simple idea is to analyze using in silico systems such as docking or that allow to rank the interaction between a substance and a target. And the other thing is to study. We have selected to working on untranslated region of mRNA for a simple consideration that we consider that DNA as the storage. mRNA and regulation of mRNA by non-coding RNA is the pathway that allow to assist a biological system to adapt their behavior and their function to a modification in environmental context. Then microRNA are one of the, and non-coding RNA are the first line to sense the environment and to change the function of the functionality of metabolic pathway without opening the genomic encyclopedia. They cast the number of regulatory signals in 3' OTR is equivalent to identify what are the combination of elements that allow to stabilize and destabilize a specific mRNA. On other term, the idea is to study with a combinatory approach in which way a specific combination of signals can increase or decrease the stability of mRNA. I do not know how many of you know the RNA surveillance. The RNA surveillance, RNA decay, are all the system that make the quality control of RNA. And the RNA decay is the multi-enzymatic system that proceeds to release of the degradation of RNA. The RNA surveillance is more important when, for instance, Professor Pasadani talking about ribosome and other. RNA surveillance must control how ribosome, the quality of mRNA and how the ribosome work. Ribosome, I was surprised, is a very complex structure with a channel in which mRNA is inserted. There are some specific sites that proceed to modify the RNA before to start with the translation. Then the problem to identify what are the signals that are required for maintaining an RNA for a specific time interval is very, very important. The other is another problem that I would like to underline is the folding of the 3' UDR. This is come back to the structural genomic approach and the modeling, because if for protein I can have a relatively large set of structure for the RNA, for the DNA, the number of information, structural information is relatively small. And as an additional problem, a protein are mainly, the structure of a protein is mainly obtained by crystallography. The structure of DNA or RNA is mainly RNA is obtained by NMR. What is the problem? The advantage is that I can obtain a structure for a short molecules. But the disadvantage is that I have several different metastructures. Because if you open any NMR entry on PDB, you can find that there are different conformations. And what conformation one select to proceed with the docking. The problem of this is to create a theoretical model that is obtained by superposition of this model, checking, evaluating, having the distances between models that is possible to attain, and then weight the docking according, weighting the results according to the parameter. It's not easy, and because the distances between the metastructure are very small. And in terms of computational science, the idea to automatize the process to create a prototype of NMR model, it's a great challenge. I am not an expert of NMR, but actually the problem is to obtain an optimal theoretical model or merged model of this metastructure. And the another thing is always related to the mRNA and the folding of the RNA in general sense, is for instance that ribozyme or riboswitch are structure. This structure has been obtained by NMR. Then when someone attempts to design antibacterial peptide, you are working on different metastructures, and you need to select one of them, or you attempt to overlap with the structure. Otherwise, changing some angles, changing some in the conformation, your peptide could reduce the binding efficiency or not. This is, in general, as I said, the problem of folding of nucleic acid is very big, mainly in the RNA. RNA is a world apart because many start from DNA, also I, and at the end of the history, the true regulator is the RNA. Don't forget that on these systems, on this messenger or ribosome, some actions are performed by other RNA. Then how to discriminate and the single step of the pathway is very, very complicated. In terms of exposome, this problem of RNA is associated with the increase of increased internal concentration of pollutants. In the slide about exposome, we're talking about the key event or point of POD, that are those concentrations which below that, below it, there is not effect. Over it, there is an effect. Or key event. The key event, we can. The key event, in any case, is a molecular event. It's completely unapparent for our life, or for from the pregnancy of your mother. But staying in a polluted way, having a bad lifestyle, using drugs or what you want, you can start to stimulate the system, start to alter the system. For instance, now I am working on phthalate, that is also in the PM10 mixture, and the phthalate are essentially working from pregnancy to age, with different impact. But there are some problems about the fertility. There are some problems about, and phthalates have a gender-specific activity. In one case, decrease the fertility of sperms. On the other case, induce some problems on the pregnancy and the fetus development. Can modify them. It's very, very, very. It's not in this example show you that for some specific situations, some specific pollutants, it's important also define the ethnic or gender different action, because otherwise we generalize into a weak association. This is another thing. And in any case, this event at molecular level, and mainly on the post-transcriptional regulation, in which are involved non-coding RNA and other molecules, is a critical point. Because they are in the cytoplasm mainly. They work in the cytoplasm. Inside that cytoplasm, there is a lot of pollutants for access or in the environment. They can start to modify all the biochemical pathway. You can see the effect after 20, 30 years, 40, 90. This is the aim, the final aim, the real aim of exposome. Screening, having a longitudinal control and a longitudinal analysis along the time axis of the different way of exposure and the different way in which an exposure interacts with our body. Okay, then I guess to have a show two possible some possibility to integrate genomic functional, two different kinds of integration. The first is strongly related to molecular biology data and experimental data. On the opposite side, we have an example of application in which we start for clinical data. For mercy, I forget to the use of social data or political data because it's not possible to consider what kind of valuable information they give. But this is the two starting point for an exposome analysis. And for my experience, I prefer to start from molecular because you have, but it's personal point of view, because you don't have the problem to spend days with the database manager or software manager of the health department to explain in which way the patient are coded or in which way we can anonymize the patient. The problem to achieve to obtain the data about clinical data are very complicated because you need to have a lot of permission. In this perspective, the exposome has a limit because I cannot access to all the data. An exposome project cannot start without the support of a governmental organization or authority. You cannot go in a hospital and say, okay, give me the information about this area. No. And the other thing is how long is the time to obtain the authorization. I understood that India is not so fast like for decision like USA. It's more similar to Italy. In order to have an authorization, we spent 6, 7, 1 year to have sending to local government, communicate, central government, so on. Similar. Then the problem to assess the data is another limiting factor. And in the design, solving the bureaucratic problems before to go on a keyboard and programming some database something is necessary. Otherwise, you can stop at some level. You cannot complete an exposome level. On the opposite, if the government is an example, I told you this example. When the Indian colleagues coming in my town, they have found there is a chimney of steel plant with carbon. And said, okay, this chimney is capable to pollute 25 square kilometers. And okay, working on the data of Koreili area near Nagpur, because they have surveyed it, the survey was carried out in this area. And I asked to, well, we can attempt to start up a bilateral project with my regional department and someone of Maharaj, O'Neill, what you want, to check the difference of impact of PM10 or air pollutants generally on the population. We have two different ethnic population. We can check, for instance, the difference of incidence of disease in my region and in Nagpur with different ethnic lifestyle, food, and life and habits different, completely different. That could be a good starting point to strengthen cooperation and to find something at higher level. The regional government said, I am not I am not interested because the next 2 weeks the next month there are election day. We know that we have many debt for powder, for dust. Normally, when talking about Bhopal, about other India's polluted, near my town there is a river that has a chromium-6 concentration 10 times higher than the average, because there was a plant to produce coal. The last summer in northern Italy, the drought has reduced the water, the level of the water, groundwater for drinking, and the last level was contaminated by pesticide. We are talking not Nigeria or what you want. We are talking officially with a high developed country. Then the problem is global, and the exposome could be, I guess, could be a good approach to break the barrier and to attempt to put together different environment, different expertise, because the expertise here are not the expertise in Italy for environment, in order to obtain an harmonization of the tools. This is my last considerations that related to this kind of tutorial. And I would like all to finish to focus the relevance of interaction between small organic molecule and nucleic acid. For the DNA is well known. For the RNA is really to do, is a challenge. I guess. Thank you very much and stay in time. This time I have the watch. Do we have some questions?